Okay, let's start with our usual question. All right, so overall, okay. You're still not daunted by the new semester? That's good. Let's ask the same question in 12 or 13 weeks from now. Hopefully you will be as excited. Everybody can see this?
Yes.
Okay, so I just wanted to bring to your attention two editorials from last week. And as we said at the beginning of this course, we are really dealing with open questions that the scientific community is grappling with as we speak. So this is from Nature, last edition. Open science done wrong will compound inequalities. So basically the idea is that people from rich countries, researchers from rich countries can afford to do the open science, but researchers from third world countries cannot. And open science is going to build a bigger gap and inequalities. So interesting read if you are interested. Another topic on the right, this is from Science from last week, and this is about the plagiarism. No one else than the Vice Chancellor at the University of North Carolina basically copied stuff and basically had to step down. So it's really quite...
Someone is trying to get into the Zoom.
So. Okay. Just a minute. Okay, sorry about that. Okay, so as we'll talk about scientific misconduct, you will be surprised how much of this actually is happening. And look at this. This is a vice chancellor. That's the second to command of a whole university. It's like Arias Tzaban deputy, basically, who did plagiarism and had to step down. So we'll talk a lot about scientific misconduct in 2 weeks from now, but this is happening and you should be totally aware of this. And when you read papers, you have to be careful. And definitely you don't want to use practices like this because the end result is really terrible. So people basically lose career over such things. And I ask you guys to read 2 papers for today's lecture. So first question that I want to ask is, did you read the Watson and Crick paper? Yeah, I hope that all of you did. So it seems maybe not all of you, but the guys who did not read, guys and ladies who did not read, don't disclose. I think that every scientist should read this paper at least once. All right, I think most of you read it. So in fact, it's not one paper, it's a story of 3 papers. And there were in this issue of Nature in 1953, there were 3 papers published back to back. That was the agreement between the 3 groups and the editor of Nature at that time to publish those 3 papers back to back. The first one is by Watson and Crick, the structure of deoxyribose nucleic acid. And that's the first of the 3. This is basically, just look at this. This paper is 1 page and a little bit. So in 1953, that was not uncommon to publish 1-page paper. And obviously, the impact of that 1 paper is enormous. Okay, the second paper is this paper, molecular structure of deoxy-pentose nucleic acid. The first one, of course, is by Watson and Crick. The second one is by, just a minute. So the second one is by Wilkins, Stokes, and Wilson. And it has this very famous picture. We'll talk about it. And the third one is by Franklin and Gosling. And this is a longer paper. It's 2 pages. But you all know only about the first one. You don't know that much about the other 2 papers. So the question I want to ask is, why only Watson and Crick paper is so well known? And basically, all 3 papers deal with the same topic, really. More accurate. First on order. Well, that's a decision of the editor, not of the people who wrote those 3 papers. Well described, more accurate, detailed description. They show other mistakes. But they show other people mistakes, but they don't show the mistake of these other 2 papers that are back to back. Most important finding. I would argue that the actual experimental data is the most important one. Easy to read, better in marketing. Understandable. Okay, well, we get all kinds of answers. Let's go over this. So what I'm going to do, it's not my own work. I'm going to use annotated version of this paper that can be found. I don't know if it's still on display, but this could be found in San Francisco Science Museum called the Exploratorium. Actually, I have very fond memories of this place 30 years ago. When my kids were young, we used to go there very often. It was the first hands-on science museum in the world. Very, very excellent museum. And they had an exhibition on the double helix. And they take this paper by Watson and Crick and analyze it. And that's what I'm going to display today. So we are going to go through all these comments that you see here in red as we go through the paper. So first of all, let's talk about Watson. Watson was a young scientist. He was just 23 years old. He did his PhD with Luria. Luria and Delbruck, very famous biophysicists, were part of the phage club at called Spring Harbor. many years later, Watson becomes the head of that laboratory called Cold Spring Harbor. Immediately after the Manhattan Project, the end of the Second World, scientists were looking for the next big thing, and they were really relying on Schrödinger, a very famous book that I think I've shown you on our first meeting, What is Life? Basically, they solved the problem of atomic bombs, and now they can go back to civilian questions. And one of the biggest puzzles is really what is life all about, and what is the genetic code, and what is the role of nucleic acid in all of this. And it was clear for the whole community that probably the best tool to do this is X-ray crystallography, because the Cambridge lab, the MRC lab, started to produce, structures of proteins, and they wanted to solve the structure of the genetic material with X-rays. So Watson decides to go to Cambridge for his postdoc, and he meets this physicist, Francis Crick, who was a crystallographer, who worked with Bragg and basically later on proteins. They were fully aware of how important this work is, and they were very competitive. And they, within the first meeting, once Watson lands at Cambridge, he started to talk about how to go about solving the structure of DNA. So they were timely. They had the right tools, so they were really primed to do this. And by the way, they were very competitive. There was another group at Caltech here in California, in Los Angeles, Spaulding, who was also on the same problem, but he had some difficulties, and we'll talk about this why. So basically, this date is considered as the beginning of molecular biology as a discipline. It was really a revolution. Until then, there was kind of a competition between two approaches to biology, vitalism versus reductionism. As I mentioned, the Phage Club at Cold Spring Harbor was a bunch of physicists coming and trying to decipher questions in biology. As physicists, they were reductionists. They wanted to have the hydrogen atom of biology, and they started with the simplest form of life, which is basically a virus, a phage that infects bacteria. It was called Phage Club. And one of the first questions they came up with, what is the structure of the genetic material? The other approach was vitalism. And vitalism basically was, you can either solve biology bottom up, or you have an up-down approach, which is vitalism. Basically, vitalists studied whole organisms and viewed genetic as too complex to understand fully. Reductionists wanted to decipher fundamental life processes as entirely possible and critical to curing human diseases. Obviously, in this paper, April 25, 1953, solved that tension between vitalism and reductionism. The reductionism won in a big way. So, as I mentioned already, Linus Pauling was kind of trailing behind Watson Crick. He also worked on the same problem. The only reason he really did not beat them is that he could not travel to to London for a scientific meeting. Anybody knows why he couldn't do that? It's actually written here. Open the mic and answer, if you can. Anybody knows what this House, an Un-American Activity Committee?
No, we are not aware of it.
Have you heard about McCarthyism? So immediately after the Second War, the world is divided into two. You have the Soviet Union and you have the US. And in US, they hate communists, and in Soviet Union, they hate capitalists. And the world becomes very polarized, like nowadays with the Ukraine war. The Iron Curtain is put up, and this former general in the US Army called McCarthy is a Congress person, and he starts a witch hunt against communists. Obviously, many Jews, many scientists were a target. Linus Pauling was pacifist. He was against the Manhattan Project, against the A-bomb, especially after they threw the bomb in Hiroshima and Nagasaki. Many scientists kind of became pacifists, but he was actually very active, and therefore he was accused to be a communist, and they took away his passport, so he couldn't travel to London. And this is possibly why he missed solving the DNA structure. Okay, so this is point number 2. Point number 3, this is Nature. Nature was the most important journal at the time, even today. It was founded in 1869. A really interesting point, we talked a lot about peer review and open science and open peer review and so on. In 1953, there is no peer review. You send a paper to the journal, the editor reads it. If he chooses to do so, he can send it to some of his friends to get some consultation. But there is no official review process. And then he makes a decision if to accept or not to accept the paper. But it was so much easier at that time. And we talked about Pauling. So Pauling was really, he actually came with the first model for DNA. He had some data. He proposed that the structure of nucleic acid is a triple helix. But because he could not travel, he did not have access to the newest data from Rosalind Franklin, like Watson and Crick had. And that's what really made the difference. So in their language, you read the paper, we wish to put forward a radically different structure for the salt of deoxyribose nucleic acid. They claim it's radically different because it's not a triplet helix, like what basically Pauling said. The radically different is strongly set it apart from the model proposed by Pauling. This claim was justified, while Pauling model was a triple helix with the base sticking out. It was basically what we call nowadays B-DNA, where if you over-twist DNA, the bases, the hydrogen bonding between bases, the pair is bright and basically bases pointing out. He had this model of three strands that are basically twisted, and then the bases are sticking out. That was obviously the wrong model. Both chains follow right-handed helices, but owing to the dyad, the sequences of the atoms in the two chains run opposite directions. So the central description of the double helix model still stands today, a monumental feat considering that the vast majority of research findings are either rejected or changed over time. So here in 1953, they say that the DNA is a double helix. It has two strands, and the two strands have opposite directions, 3' to 5' on one strand, and then 5' to 3' on the other hand. This model is correct, and it's, as it says here, it's still today correct. It was never refuted. Obviously, DNA comes in many different structures if you go to some extreme conditions, but the way DNA is packaged in our cells is in this fashion. So science historians, for example, McElney and others claim that this discovery stands on the same pedestal as Newton and Heisenberg and Schrödinger and Bohr and Kepler. This is one of the most fundamental findings in science till today. It enabled solving the Human Genome Project, and later, together with CRISPR-Cas9 technology, we will probably solve genetic diseases as well. So this all started with this discovery of the double helix. Now, as we talked about the scientific method, obviously they had to rely on a lot of information that was gathered and processed before they attacked this problem. And this Sweden scientist, Furberg, already had a DNA model. He already kind of figured out the issue of base pairing. And they heavily relied on his work as well. An interesting, there is one figure in this paper, and this is what you see here, figure number 1. And this figure was basically drafted by Crick's wife, Odile, I don't know how you pronounce her name. But at that time, we did not have PowerPoint or Illustrator or many other tools that people are using. And I remember even for my own papers at the beginning of my career, we drew some figures by hand. So once they solved the structure, they actually build a model from cardboard to make sure. And once the model kind of satisfied the constraints from the X-ray diffraction that Rosalind Franklin measured, they were convinced that this is the correct structure, and they asked Odile to basically draw this, and this is the figure that is accepted for this paper. The last hurdle that they had to figure out was the base pairing of the 4 bases and how they distort the helix. As I said, they basically made a model cutouts from cardboard and tried to arrange the bases. You know that one pair has 3 hydrogen bonds and the other pair has 2 hydrogen bonds. And basically they were playing with the cardboard to satisfy the sugar chains, the side chains, and the base rearrangement with the pairing to get this right. And basically, till today, many chemists have in their drawer sticks and balls, and you build models. Nowadays, you have structures in PDB, you have 3D printers, and you can actually print your models and so on. But it used to be very common that you will have a set of balls and sticks, and you would build model out of them to solve structures. So as I mentioned already, it's not really one paper. Those are 3 different papers. They're all talking about the same topics. They all contain and refer to the same data. So basically, Nature publishes 3 papers, one by Watson and Crick, one by Wilkins, and one by Rosalind Franklin. The Wilkins and Franklin papers describe the X-ray crystallography evidence that helped Watson and Crick devise their structure. The authors of the 3 papers, the lab chiefs, and the editors of Nature agreed that all 3 would be published in the same issue. The following communication that our authors are referring to are the papers of Franklin and Wilkins, published on the journal pages immediately after Watson and Crick's paper. So let's read what they're saying here. So far as we can tell, it is roughly compatible with experimental data, but it must be regarded as unproved until it has been checked against more exact results. Some of these are given in the following communication. So basically, they refer to Rosalind Franklin data as something that happens after they solve the structure. They come up with this paper, they have a structure, and they say, Rosalind Franklin data basically is in agreement with our model. they don't really say anything about how they arrived at this conclusion and if they had any chance to look at this data. The impression they give, really, they don't give any credit for the people who took the data. So it's all their doing, they solve the structure, and somebody else does experiments, and maybe they agree with what we have done. We were not aware of the details of the results presented there when we devised our structure, which we trust mainly, though not entirely, on published experimental data and stereochemical arguments. They go further and they say, we did not have access to this data. Our solution of the structure of DNA comes from basically other previous published data and our cardboard model, where we try to move things around and figure out what works and what doesn't. And guess what? This is a blatant lie. This is not correct. They don't account in this paper the correct historical sequence of events, and they don't give credit to the people that deserve credit. So this sentence marks what many consider to be an inexcusable failure to give proper credit to Franklin. So there is really a very, very sad story about the whole thing. These two guys got a Nobel Prize, and Rosalind ended up, dying from a disease at a very young age, not getting credit for the work she did. Watson and Crick are saying here that they were not aware of Franklin's unpublished data, yet Watson later admits in his book, The Double Helix, that these data were critical in solving the problem. Watson and Crick knew this data would be published in the same April 25th issue of Nature, but they did not formally acknowledge her in their paper. What exactly were these data, and how did Watson and Crick gain access to them? While they were busy building the models, Franklin was at work on the DNA puzzle using X-ray crystallography, which involved taking X-ray photographs of DNA samples to infer their structure. By late February 1953, her analysis of these photos brought her close to the correct DNA model, but Franklin was frustrated with an inhospitable environment at King's, one that pitted her against her colleagues. And in an institution that barred women from the dining room and other social venues, she was denied access to the informal discourse that is essential to any scientist's work. So basically, Franklin was on the way out. She was in King's College. She was a female scientist, looked down at, could not go to the dining room with a man. Can you imagine? And basically, people did not talk to her. She could not get information and discussion and brainstorming with colleagues. She was miserable. She decided to leave. Seeing no chance for a tolerable professional life at King's, Franklin decided to take another job. As she was preparing to leave, she turned her X-ray photographs over to her colleague Maurice Wilkins, a longtime friend with Crick. Then, in perhaps the most pivotal moment of the search for DNA structure, Wilkins showed Watson one of Franklin's photographs without Franklin's permission. So basically, her colleague Wilkins takes her data, shows it to another competing group without asking for her permission, and she doesn't get credit. She took the data. You cannot imagine something like this happening today. If you're writing a paper that is based on somebody's data, you either bring him in as a co-author, or at least you give some credit. So that data was given to Watson and Crick without her permission. As Watson recalled, the instant I saw the picture, my mouth fell open and my pulse began to race. To Watson, the cross-shaped pattern of spots in the photo meant the DNA had to be double helix. So they actually did already computation, and they had predictions. They basically had a hypothesis, and they knew what to look for. In their view, based on simple Bragg theory. If you have many double helixes in a crystal and you shine X-ray on them, you should get a diffraction pattern that looks like X. This is a famous photo 51 that I showed you before. And the minute he saw that picture, he knew that their double helix is the correct solution.
Shimon?
Yes.
Can I, like one of my professor, when like we were discussing this paper in my master's,
Yes.
she said that when the Watson and Crick get like the like acknowledged for this paper, they acknowledge Wilkins about like, you know, its contribution to the structure, but not the Rosalind, which like the main data come from the Rosalind Franklin X-ray crystallography.
Well, they don't even acknowledge Watkins.
Yeah. But one of my professor gave the reason because at that time, female scientist was not appreciated or something.
Yeah, that's absolutely true. And she couldn't walk into the dining room in King's,
that was
which is unbelievable.
Okay.
So was it unethical for Wilkins to reveal the photographs? Should Watson and Crick have recognized Franklin for her contribution to this paper? Why didn't they? Would Watson and Crick have been able to make their discovery without Franklin data? So this is all open questions, and it really, I think, sharpened the whole issue of misconduct and ethical behavior as scientists. Obviously, they won glory. They started a whole new field. Not to minimize what they've done tremendous. I mean, still going from the X-ray structure to the actual model was a stretch to gap, and they did that. But not giving the credit to the people who took the data, it's just unheard of, unbelievable. And, you know, we have this type of open questions ever since. Obviously, this is not one, you know, behavior we want to encourage for scientists. So very, very sad story. And then maybe the most important sentence in their paper: it has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material. So this phrase and the sentence it begins may be one of the biggest understatement in biology. Watson and Crick realized at the time that their work had important scientific implications beyond pretty structure. In this statement, the authors are saying that the base pairing in DNA provides the mechanism by which genetic information carried in the double helix can be precisely copied. Knowledge of this copying mechanism started a scientific revolution that would lead to, among other advances in molecular biology, the ability to manipulate DNA for genetic engineering and medical research, and to decode the human genome along with those of the mouse, yeast, fruit fly, and other research organisms. So that was really a start of a field. And the last comment here is what they said in the next paragraph: full details of the structure, including the conditions assumed in building it, together with a set of coordinates for the atoms, will be published elsewhere. And that was a common practice at that time. So it used to be that you publish a very quick letter, one page as you could see, and then follow up with a longer paper which has all the details. What nowadays we put in supplement information, SI in papers, they used to publish as a second paper. So that's this paper. Another interesting read, I'm not going to read anything here, I just give you here the reference if you are interested. It's a very different take on the birth of molecular biology. This is essay by Halloran. This is basically an essay in the rhetoric criticism of scientific discourse. And it really talks about the ethos of this paper. So it's not a scientific paper, it talks about the ethos that was built following this paper. So if you're interested, you're invited to read this. So this is a paper that is written 50 years after by Tobin, and he drew 3 different lessons from the whole story. First lesson, collaborating with faculty colleagues is more profitable than competing against them. Second lesson, research has no value if unpublished. So basically, Rosalind Franklin did not publish her data. She was sitting on it and not publishing it. But she was busy. She was kind of moving from one place to another. And third lesson, Watson and Crick have something important to say. They say it clearly and then shut up. The article exemplifies the 3 cardinal qualities of good scientific writing: brief, clear, and simple. So as I showed you, their paper is 1 page. Franklin is 2 pages, Wilkins is 2 pages. And when you read, you know, their paper is really very simple to read. It flows very well. It's logically built correctly. It has 1 message. It's precise. And all of the reasons that we gave in the past 20 minutes really are the reasons why this paper is so much more well-known than the 2 others. Although the actual paper with the data, in my view, is as important. All right, so that's 1 paper. Any questions? Any comments? Any thoughts?
As far as I understand, they had, I mean, they didn't gain the information in an unethical way. The only unethical way they think they did is just don't give credit in the paper. Not enough credit, I think.
Yeah, you're right. I mean, Wilkins shared the information. If there was an unethical act, it was Wilkins sharing the information without permission. Right? That's an unethical act on his side. On their side, they should have acknowledged this. They clearly lie in the paper. They say in the paper that they did not know about this data. That was not true. They got the data way before they published the paper.
Yeah. Okay. Thank you.
Any other comments?
Yes, I found it weird that they didn't share any of the calculations. I think that if they shared them, they would have had to say like how they jumped from here to here.
Sorry, I cannot hear you well. Can you speak closer to the mic maybe?
Yeah, can you hear me now?
A little bit better.
Wait, I'll just check.
Okay, this should be better.
Okay.
So what I found weird is that they didn't share any of the calculations, like how they actually proved that this is the right way.
So I commented on this. It was common practice to have a follow-up paper where they do share the calculations.
So they do share it just separately.
In 1954, they have a long paper with all the details.
Okay.
So what you would put today in an SI was put in a different publication at that time.
It might be a little bit off topic, but I found it very strange and rude there, because in the beginning they mentioned the 2 papers and they very rudely said that it is wrong, and in one point they even said it is so wrong we are not going to comment about this.
No, you're talking about the reference to polling.
Yes.
Okay.
It was very strange to see this kind of criticism with peers in the same scientific.
So that was common practice at that time to attack.
Okay.
we have become much more politically correct over the past 5 decades, but people were not ashamed to say that somebody is wrong, and obviously polling was wrong.
Okay, it's fine to say this paper is wrong, but the way they said it, I just found it to be
the
very
style.
rude.
you're talking about the style.
Yes.
Yeah. So
you
the
can
style.
say anything, but with a little bit of.
So the competition and the rivalry between scientific groups 50 years ago and the language, the political correctness, it was a different story altogether.
Okay.
Just to show you how we evolved. It's not only politics and all kinds of other things, but it's also in science. It's just the way we conduct ourselves has evolved. But yeah, you would attack people you thought that they are wrong, and you would put it in paper and you would say, you know, loud and clear. That was the practice of the day. Okay. Any other comments, thoughts, questions?
Yeah, I have a question too. In the second paper, at first I read the second paper that was attached to the first paper, at least the half of it was attached. And we can see an X-ray image. Doesn't it raise suspicions that they published now the X-rays and Watson and Crick didn't?
So I think they did here a nice acrobatics, right? Because they come up with a structure and they say, this is the picture, right? The minute they saw it, they knew it's double helix, because that, you know, they had some limited computation, but the computations, and they tried helix as one of the possible solutions. So they knew when they saw this X, that's the famous photo number 51, they knew it's double helix. So they had a problem. They wanted to publish their solution to the structure. And they basically say, we just use other people's data, not Franklin, and we use cardboard model, and we solve the structure. And other people's data, now they refer to Franklin, which is following our paper, agree with what we came up with. That's their logical argument.
Okay, and I have a question.
They basically clearly make manipulations to say we have not built our model based on Franklin data. That's what they say in the paper.
Okay, and Franklin did not know how to interpret this?
Well, the claim is she was pretty close. Moreover, she did meet with them before, because as I said, Wilkins was a friend of Watkins. And so they met. You know, Cambridge, London is 2 hours. And they had some meetings, and early on, Watson and Crick came up with the wrong model, and they had a discussion, and Franklin said, no, it cannot be. They had to do with some water molecules, you know, attached to the nucleic acid or whatever, and their initial model was not correct, and she was able to tell them why the model is not correct. So she was close. Maybe she did not have the same math skills, she was not as advanced in solving, but she was close. You know, the most logical thing to do was, since those groups did talk to each other, was to come together, collaborate, and publish together. That's what I would do. But there was a competition. Watson and Crick competed with Pauling, and for some reason they did not, you know, consider Franklin as somebody that they should really consider as a co-author or whatever. And they just went, used her data, said that they did not use her data, and published the paper. So I find this to be unbelievable and sad, and but this is how things, you know, played out.
Thanks.
Okay, let's move to the next paper. So the same question.
Does reading include comprehension?
No.
Okay, good. I read it then.
And thank you for being honest. This is a much harder paper to read. It's not written as clearly as Watson and Crick, and moreover, it's translation from German, and the translation is not great. So the read is more difficult. Okay, so the reason I asked you to read this paper is this is really a segue for next week, which I'm going to dedicate for my own research. We are going to discuss this paper by Theodore Forster, Energy Migration and Fluorescence, that was published in 46 and then in a different version in 48. By the way, Forster did not denounce the Nazis and actually got promoted during the Nazi era. So this is actually a controversial issue. We call FRET, fluorescence resonant energy transfer, also Forster resonant energy transfer. And some people in the community wants to stick to fluorescence resonant energy transfer and not mention his name just because of his past. So here are the two versions of the paper, the 1948 paper in German. This is a translation that was published in 2012. It's really amazing that until 2012, the original paper was not translated. And that was published in the Journal of Biomedical Optics. So I'm going to, again, rely on the shoulders of other giants when I analyze this paper. And here I'm using the analysis of this paper by Robert Clegg. The late Robert Clegg was a professor in Urbana-Champaign, and for many, many years, he was, before coming to Urbana-Champaign, he was in Germany, in Göttingen. And he wrote a book about fluorescence resonance energy transfer. I knew Robert Clegg, a very, very nice guy who did fantastic work on FRET. So Forster theory and the accompanying experimental work on energy transfer is the most widely known and most influential of all FRET publications. Forster provided an accessible theory in a form that was practical for experimenters. It is difficult to exaggerate the influence of this work. The extensive, widespread use of FRET in physics, engineering, chemistry, biology, and medicine are due to the description of energy transfer given to us by Forster. So Bob Clegg wrote this history of FRET that is cited here, and you're welcome to read this. So for those of you who read the paper, what was Forster's motivation to study energy transfer or energy migration, in his words? Well, that's the first paragraph on the introduction. I counted 16 people who read the paper. I don't think you need to comprehend the paper in order to understand what the motivation is. Can somebody help me? What was the actual physical system he's discussing? What was the motivation based on some physical phenomena that he wanted to study energy migration? He's specifically referring to, okay, that's what I was looking for. Photosynthesis. No other explanation to photosynthesis. Okay. All right. Thank you. So Forster apparently became interested in the energy transfer process because of the non-effectiveness of photosynthesis, as Oppenheimer did. You know, Oppenheimer was the head of the Manhattan Project. He was also dealing with this question. He was also aware of the work of parents. Parents were father and son in Paris who worked on type of related question to energy transfer in the beginning and the early part of the 20th century. Experiments by Emerson and Arnold in 1932 had shown that the capture and utilization of light energy by plant leaves was much more effective than would be expected if it were required that photons exactly hit the reaction center. This is basically where carbon fixation is happening. That is, that there are too few reaction centers in the leaves to explain the very effective extent of energy capture. So basically, the leaf takes light from the sun and basically taking CO2 and fixed carbon. And the reaction is way too efficient from light to fixed carbon if you count the number of reaction centers in the leaf. This is where basically the enzymatic reaction is happening. And that was a puzzle. So Emerson and Arnold did those experiments and they showed that there is a discrepancy and they really did not come up with an explanation. According to measurements by Emerson and Arnold, short-term irradiation with high light intensities, they use sparks from a discharge light source, produces saturation effect in the algae chlorella, such that the assimilation yield does not exceed a certain threshold, even at arbitrary high light intensities. This limit is reached between 2 and 4 light quanta per 1,000 chlorophyll molecules, the number of light quanta required for reduction of 1 CO2 molecule. Gaffron and Roll conclude from this that this number of molecules contribute their absorbed energy for the reduction of a single CO2 molecule. As this has to be assumed to take place in distant location, this interpretation requires energy migration from the chlorophyll molecule to the location where the reduction takes place. So based just on this imbalance between, or the high efficiency of this process, there are too many chlorophyll molecules, there are too few reaction centers. It takes basically 2 to 4 photons to do this reaction. Forster comes to a conclusion that energy has to migrate from the antenna to the reaction center in order to explain this process. Until then, people knew about energy migration in the form of electron transfer. This phenomena was well known, but here the distances did not really allow for this to happen here. It wasn't an electron transfer reaction. So energy had to migrate over longer distances, not that just by contact between atoms. Forster knew of these results and reasoned that an extremely efficient transfer of energy between the chlorophyll molecules must be responsible for the eventual diffusion of the energy, which was absorbed over the whole surface of the leaf into the relatively sparse reaction centers. So basically, the leaf as a whole acts as an antenna, collects the light, and somehow that energy collected from the light need to get to the right place to do the reaction. And how this migration happens, that was what he set up to study. He assumed that this energy diffusion is due to energy rapidly hopping between molecules. He was also aware of the earlier work of the parents and the other data indicating energy transfer over distances longer than the molecular diameter. So nowadays, when we look many, many years later, we have crystal structure. And you can see here light harvesting complexes at the crystal structure. This is from bacterial roll option, for example. This is the reaction here, and you have all these chlorophyll molecules sitting on those rings, collecting light, and somehow the energy ends up here. And for many different other organisms, you can see very similar concept. You have antennas to collect the light and somehow bring the energy to the reaction center where the chemistry happens. Another example, this is again a bacterial system, I think. You have all these rings. Yes.
Can you go back a slide? What is figure A? What are those coils?
This is one ring.
One ring.
I think it's, I don't remember the caption here, but I think it could be different organisms. So light harvesting complexes and reaction centers occur obviously in plants, in algae, in bacteria that use light. So there are many, many forms of this, but the concept is the same. You have basically chlorophyll molecules as antennas absorbing the light, and somehow the energy is migrated to a reaction center where the actual carbon fixation is happening. So there is a reaction center here at the center, and you see like 3 rings and a reaction center. Here you see 2 rings with 2 reaction centers, and so on. Does that answer the question?
Yes, thanks.
And here, in this case, you have 6 rings. However, it has to be pointed out that the existence of an assimilation unit and therefore energy migration in this assimilation unit is very controversial. So at that point, people really think about energy migration only as electron transfer, as contact. Forster then brings other non-biological energy transfer examples, synthesized photochemical reactions, concentration-dependent depolarization. This is the experiment of pairings, concentration-dependent quenching. So basically, there are other examples from chemistry experiment or physical chemistry experiment that show that there is some kind of energy migration over distances which are larger than just contact between atoms or molecules. So he adds, it is now mostly assumed that in biological processes, the energy migration without being linked to a material carrier of any kind. So that's really a key sentence in this paper, is hinting to some kind of a mechanism for energy that can go from one place to another, not by actual moving atoms or electrons or whatever, some material entity that can take the energy from one place to another, but it happens through a different mechanism. And this different mechanism is dipole-dipole interaction. So this is how your radio works. You basically have an antenna, the transducer, you accelerate electrons in this antenna, and that radiates electromagnetic waves into free space. And on the receiver side, you have another antenna, the electrical field in free space, with electrons in the receiving antenna, and this is read out by electronics on the receiver side. So he claims that energy is transferred between molecules or atoms through the same mechanism of dipole-dipole interaction. So when atom is excited, it basically has oscillations. You have Rabi oscillations as long as the excited state is still coherent before it decohere. And these oscillations basically have energy that can be propagated and absorbed by another molecule nearby. So it's called radiation-less energy transfer, dipole-dipole interaction. And if it is indeed dipole-dipole interaction, there are very clear predictions how efficient that energy transfer is and what is the distance dependence of this energy transfer. So in terms of Yablonski diagrams, the energy states of 2 molecules, fret happens between 2 molecules. We call the first molecule a donor molecule, the second molecule an acceptor molecule. This is S0, the ground state. This is S1. And you have vibronic manifold for each of those states. If you use sunlight or laser and you promote an electron from the LUMO to the HOMO, or from the valence band to conduction band, if you're talking about semiconductors or nanocrystals and so on, you now excite the system, and it can basically relax by emitting a photon and go back to the ground state, or it can actually relax through non-radiative process where you have some phonons or vibrations excited, and basically you go back to the ground state without emitting photon. If you are bringing another molecule next to this donor, which we will call an acceptor molecule, which has the same type of energy level structure. However, the gap here, the band gap or the gap between the LUMO and the HOMO is smaller. Then once you are in the excited state here, you can transfer energy to the acceptor molecule by dipole-dipole interaction. And this molecule that was initially in the ground state finds itself in the excited state without actual emission of photon from the donor and absorption of photon from the acceptor. This is why it's called radiation-less. Basically, once you are in excited state in the donor, you have a oscillating dipole, and this oscillating dipole can be received by the antenna, and suddenly the acceptor finds itself excited. Acceptor as well can be relaxed through emission of photon or through vibrations. In terms of quantum mechanical states, you have donor and acceptor in the ground state, you add one photon. Now you have donor in the excited state with the asterisk here, and acceptor in the ground state. You have a rate that is proportional to the energy transfer. It's called KT, where this excited donor and ground state acceptor convert into a ground state donor and excited state acceptor. And from this state or this state, you can basically go through radiative relaxation back to the ground state. So basically you go back to unexcited acceptor and donor and emission of a photon from the donor. If you are in this state, you can go to unexcited acceptor and donor and emission of an acceptor photon, or you can have non-radiative decay of these two states. So these are the all possible states that can happen in this configuration. So Forrester basically worked out all those rate equations that define the system, and basically could write this equation that the energy transfer efficiency, how efficient is it to move from here to here? It's basically the rate KT that you see here divided by the sum of all rates, KT plus KD plus KDI. And he showed that this is actually scaling like 1/1 plus the ratio of the distance to some physical parameter that we call R0 to the 6th power. This is a very, very strong distance dependence for this rate or transfer efficiency. R0 we call the Forrester radius. It contains many physical parameters. We'll talk about this in a second. So you refer the distance, the molecular distance between those two molecules to this parameter. It's basically the distance at which the energy transfer efficiency is 50%. The transfer efficiency also is dependent on the donor lifetime, 1 over the donor lifetime, R0 over R to the 6th power. This is another way of writing the same thing. Again, this is basically the same Yablonski diagram. You excite the donor with a blue laser, it can emit a green photon, or energy can be transferred and you are getting a red photon. If those two donor and acceptor are going to be at a very, very short distance, energy transfer efficiency is going to be very high. And this molecule doesn't really have a chance to emit a green photon. All the excitation will move to acceptor and you always get only a red light. So basically you kind of stock shifted your dye pair. Instead of getting emission in the green, you are getting emission in the red. If they are far apart, that is not efficient and you have more chances of getting this green photon than getting the red photon. As I said, this distant dependence of energy transfer is very, very sharp. It actually has this 1 minus a sigmoid shape. The FRET efficiency at very short distances between donor and acceptor is very high, 100%. The FRET efficiency of very far distances between donor and acceptor is 0. And there is a range where you have this quick change in the probability of energy transfer. And at the distance, which we call R0, you have 50% chance. But what you have here, let's say from this point to this point, you have more or less a linear dependence between Fret efficiency at distance. And therefore, this can be used as a molecular ruler. You can actually measure distances on the nanometer scale. The free parameter are not, the physical parameter are not, or the Forster radius depends on the details of the two dyes and many other parameters. We'll go over this in a second. And here you see a plot for different R0s. You see that this curve is shifting for larger R0s and also becoming flatter as you go to larger and larger R0.
So you can measure concentration that way?
Well, you can, but you need to do more things. But the main thing you do with this is you measure distances. This is how this method developed. And we'll talk next week about how you may measure changes in distances, or dynamical changes, conformational changes, molecular machines at work. But initially, and it took some time since the theory was published and until people started doing some meaningful experiments, it took more than 2 decades. But the first introduction of this concept as an experimental tool was to actually measure distances on proteins and nucleic acids, not concentrations.
But if you have a solvent of, I mean, I don't know anything about chemistry, but if you have a solvent with different kinds of chemicals, let's say the same kind, okay? And this reaction depends on the distance between them, right?
Yeah, so I think if you take the same molecule and you go to higher and higher concentration, at some point you can have fret between the same molecules. This is called homofret, and that gives rise to quenching. Okay. So you can measure the amount of homofret as function of concentration, for example, and have a calibration curve. Mm-hmm. All right, so we talked about R0, the Forster radius. It depends on 4 different physical parameters. R0 is the Forster radius. J of Ni is a spectral overlap of donor emission and acceptor absorption. We'll show a graph in a second. Phi D is a donor quantum efficiency, how good of a dye it is. N is the index of refraction of the medium. And kappa squared, that's the most difficult parameter to grasp and put your hands on, is the relative orientation of donor and acceptor dipole moments. And here we often take some assumptions. So spectral overlap is basically the overlap between the emission of the donor and the absorption of the acceptor. So here you see a pair, a fret pair. Donor is called tetramethylrhodamine, DMR. The acceptor is Psi5. In dashed lines, you see absorption curves. In solid lines, you see emission curves for these 2 dyes. Absorption and emission are always mirror image due to the Kasha rule. And if the emission of the donor overlaps with the absorption of the acceptor, you have resonance. This is why it's called fluorescence resonant energy transfer. So this is the spectral overlap, and this is given by this integral here, 0 to infinity of the normal donor emission spectrum. So you need to have this emission spectrum. This is F of D, solid green line. Epsilon A is the molar extinction coefficient of the acceptor. This is something you can measure on a fluorometer in a cuvette, which is a cross-section of each molecule for absorbing light. Lambda is a wavelength, and d lambda. So if you do this integration, you basically get this parameter. And these all are measurable quantities, so you can calculate what the spectral overlap is per given pair of donor and acceptor. The kappa square is really the dipole orientation of those 2 molecules. Usually we tend to think that those molecules are freely rotating on a very fast time scale that is basically faster than the excited state lifetime, so that we are talking about hundreds of picoseconds or even shorter. and therefore their orientation can be averaged out. But this is not always the case. If you have viscous medium, or if you have dyes which you call bisfunctionalized dyes that you can attach in 2 different points, they are no longer free to rotate, then this kappa square can get all weird values. If the 2 dyes are really freely rotating fast, then you can do the integration and average, and you find out that kappa square gets this value 2/3. So for many years, that's what people used when they measured Fred. They assumed that the 2 dyes are fast rotating. If they are not, then kappa square can take any value between 0 and 4, and that gives huge error in actual distance measurements. So this is just to some examples. If the 2 dipoles are parallel, one on top, one after each other, kappa square can get all the value of 4. If they are parallel in this fashion, it's 1. If they are orthogonal to each other, it's 0. And if it's randomized fast-moving dipoles, it's 2/3. So this is a picture we like to think. 2 dyes are attached to a protein, let's say. They have some tethers, and therefore they are freely rotating, and therefore you can use the 2/3 value. But you can get big errors in your measurement of distance if that's not the case. So the way to solve this is to actually measure the dipole orientation of your dyes, and this is done by fluorescence anisotropy measurements, where you can actually measure the dipole orientation. Then you add more constraints to solving the problem, and you can get more accurate distances. Now, how you measure Fred, there are many ways of doing it. We are talking, as of yet, just about ensemble measurements. So what we assume is that we have a cuvette with a large number of pairs, let's say proteins that are labeled with donor acceptor, or 2 proteins that interact with each other. One has donor, the other one has acceptor, and now they bind. And we want to measure the Fred, and from the Fred, we want to measure the distance. So one approach is just to do spectroscopy, and you measure the donor-only sample, and you titrate very carefully to have the same concentration of a donor-only sample versus donor plus acceptor sample. You measure the spectrum for both, and from that change in the intensity of the spectrum here, you can actually deduce the energy transfer efficiency. And knowing R naught, you can go from energy transfer efficiency into distance. So basically, you measure the fluorescence in the presence of acceptor, and you normalize it to the fluorescence with the absence of acceptor, 1 minus, that gives you energy transfer efficiency. Another approach is to use time-correlated single photon counting to measure fluorescence lifetime rather than intensity. You prepare those same 2 samples, and you go to a time-resolved fluorometer, and you measure donor lifetime, the donor-only lifetime. In the presence of acceptor, the lifetime will be much shorter because you provide more de-excitation pathways for the donor, and therefore the lifetime of the excited state of the donor will be much shorter under Fred conditions. And just measuring those 2 lifetimes, 1 minus the rate of 2 lifetimes, give you the Fred efficiency. And the last approach is actually to look at the acceptor emission and see changes in the acceptor, and again, you use this formula to extract the energy transfer. And again, if you know R naught, you can get distance from this. So that was basically the theory written in a relatively complicated way. So just to contrast, so many of you are not expert on DNA or on Fred, and you read 2 different papers from outside of your field. I guess you felt much more comfortable with Watson and Crick paper than with Forster paper, and that has to do with style of writing. We talked quite a bit last time about what is a good paper, how do you write a good paper versus not so good paper. Although the writing style of Forster is not great, the paper still had huge impact. I wouldn't say as much as Watson and Crick, but still, this is a very highly cited paper. It was very influential. However, conditions are conceivable which, even in high concentration, do not allow us associates to be present or to form after excitation, it would only be required that each chlorophyll molecule is sufficiently deeply embedded in a corresponding protein, or that it is bound at a defined position of a protein structure. However, a quantitative treatment of such a model on the basis of the theory developed here appears not yet possible for want of sufficient data. Basically, what Forster is saying here is that the chlorophylls has to be in some kind of an organization with respect to the reaction center. This is an amazing insight in 1948, 46 or 48, compared to the first crystal structure of what I've shown you before for bacterial reaction centers, light harvesting complexes. Those structures came in the 2000s and maybe even later. I don't remember the year. But basically, 50 years before the structures are solved, he says chlorophylls have to be embedded in proteins in a certain way to ensure this efficient energy transfer from the antenna to the reaction center. A final clarification of the mechanism we are dealing with here will probably only be possible on the basis of a more complete knowledge of physiochemical structure of the basic matter involved in it. We only wanted to demonstrate that this type of energy migration in a non-biological system is more frequent than generally assumed, and that by and large, it is a phenomenon which is theoretically understood. We now have an overview of the conditions for its occurrence. Where these are fulfilled in biological system, energy migration has, of course, to be taken into account for the interpretation of the observed effect of light. So basically it says, we need to solve the structure and then we can do an accurate computation. And guess what? Obviously, after the structure was solved, people were able to do very accurate calculation. And in terms of donor and acceptor, in all those systems, we don't have one donor and one acceptor molecule. We have many, many chlorophylls and one reaction center. So the computation is more difficult, but all these computations have been done after the crystal structure was solved. It's basically the same phenomena, but the math is a little bit more complicated. So to summarize this Forster paper, Forster theory is the basis of our present understanding of FRET and most of its applications. Within the validity of this model, which encompasses most of the applications, Forster's original theory still applies. The theory has been tested thoroughly, and is the conditions for which his derivation is applicable are met. The theory has always been found to be valid. The original theoretical description of energy transfer set the stage for subsequent application of FRET in many fields of research, from pure physics to biology, and it is his theory that still is used to interpret experimental results. He extended the original idea of pairings involving the well-known interaction of molecular dipoles. However, his insight and great contribution was to provide the quantitatively correct and very practical description of the FRET process in terms of experimentally accessible parameters. And FRET has been shown to be broadly applicable and extremely informative for determining molecular interactions and to measure molecular distances that are impossible to determine otherwise. All right, so let me stop sharing and ask you if there are any questions.
I kind of lost you with the second paper.
Okay.
The paper was very hard for me, but the explanation as well. And I didn't really understand the main point regarding this paper.
The main point is that if you put two molecules one next to each other, they can exchange energy by dipole-dipole interaction. You excite one molecule and suddenly you find the other molecule excited and the first one de-excited. And there was no material going between those two molecules. So think about the radio. You broadcast and you accept, and now you're excited.
This is clear, but this is not the subject of this course. So I didn't understand.
Okay, what is the subject of this course? I asked you, I think the first question I asked, what do you expect to learn from this course?
I wanted to criticize this article, and despite the fact that it's incomprehensible for me, that might be related to the field and my knowledge and my abilities, but beside that, I don't know what else.
So I think that Watson and Crick is a good example for a well-written paper with a very important scientific result. I think that Forrester is an example for not so well-written paper with a very important scientific result. So these are the two examples. Does that answer the question?
Completely, yes, thank you.
Okay. So sometimes you can have a very influential paper that is not well written. This is an example. And by the way, this is a segue for what I want to tell you next week. So, other questions?
You said about this paper, then it's like bad writing.
I didn't go that far.
No, okay. But like, it's how you can know if not because it was translated English, like maybe German.
I think part of this is translation. But in general, I think the German writing is more cumbersome just because of the language. And scientific, and German scientists are usually tend to be very thorough, very methodological. They present, their arguments and the data in a very dry way. So we are talking about style as well. We are talking about schools of how you write papers, the German school versus maybe the Western type of writing. So I wouldn't say it's bad writing. I think we are not used to that type of writing and reading. It's harder to read. That's a fact. I guess if you are a native German speaking and you read the original, maybe it will be easier. But still, I suspect it's even then a hard paper to read. But it is extremely important paper. Again, I wouldn't put it on the same level as Watson and Crick, but it is an extremely important paper.
But how can we compare this paper, Forster paper, to Watson and Crick? Because Watson and Crick didn't publish here all the results and their calculations. It was only the idea, the hypothesis.
Because they were afraid to get scooped.
No, I understand why they did that. I just say that we can't compare.
Yeah, look, of course. And I just wanted to give you examples. It's really comparing apples and potatoes, whatever, oranges. It's not the same, of course. I don't deny, but we talk about concepts. We talked about good writing versus bad writing. We talked about open science. We are going to talk about misconduct. These are two important influential papers. They are not similar. They are different fields. The format of the papers are very different. The style of writing is very different. Those are examples. We are collecting examples. At the end of the day, we want to kind of have maybe rule of thumb for what's good, what we are going to try to mimic, and what we are going to not to try to adopt because we think it's not good. But the fact is, he had insights to what is the actual mechanism of energy transfer in the leaf. Light is absorbed in the chlorophyll and gets to the reaction center, and that mechanism was not clear. Moreover, there was a big puzzle. People did not understand. When they did the stoichiometry and they measured how many antenna molecules there are and how many reaction centers, and that meant that energy has to go from one place to another somehow, and they did not understand how. He came up with an explanation. And probably he could have written that paper in a much clearer way. More questions?
Why Watson and Crick still have the Nobel Prize for this paper, even though that there are like so much controversy over it?
Well, the controversy is not about the actual finding.
Yeah,
Controversy is about the conduct, not about the finding. They were the first to come up with a double helix, period. That's a huge achievement. And that opened the way to understand how the genome is being replicated. That started molecular biology. So there is no question about their contribution. The question is about their ethical behavior, not about their contribution. Contribution was enormous. We'll talk a little bit about the central dogma of biology next time.
Okay.
You know, for the way biology works, basically you have the genetic code in DNA, you transcribe to messenger RNA with an enzyme called RNAP, RNA polymerase, my group is working on. And the messenger RNA is basically, this is the instruction tool for the ribosome to make proteins. The minute the double helix structure was solved, it was clear that they should find an enzyme that separates the two strands and replicate each strand. That's the DNA polymerase. And, you know, each of these enzymes eventually got a Nobel Prize, and sometimes more than one. So we'll talk a little bit about this next time. But, you know, it started everything we know today about molecular biology, biotechnology, medicine. It was huge, huge discovery. So we don't take that away from them. We just say they could have handled the Rosalind Franklin episode a little bit better and be a little bit more honest. Eventually, in his personal memories book called the Double Helix, and I showed, I think, the cover last time in the first meeting, he admits that he got that data before they published the paper and they solved the structure. Too late. Too little, too late. Other questions? All right. So we'll meet next week. And next week is kind of fun for me because I'm going to give you overview of things that we do in my group, we've done over the past almost 3 decades, related to Fred. And the last meeting in 2 weeks from now, we are going to have a really interesting case to study about misconduct. And following the Passover vacation, we are going to hear from you guys. I actually have not had a chance to look at the Google Sheet. Are you filling up the slots and everybody has a date? Leon, what is the status on this?
We have some full lectures of talks and some are not. So.
Okay, and I got only few people asking me to approve the topics. So I expect all of you to send me suggestions. Make sure that you're not picking somebody else's topic. So the people who have not done so as of yet, please pick up a date and pick up a paper. Okay, guys. Thank you so much and good night.
Good night.
Bye, everybody.
Good night.
Nice.